In this video, we will look at growth of our Streptomyces venezuelae colonies and assay them for production of the antibiotic chloramphenicol. Let's get our plates out of the 30-degree incubator and have a look at how they've grown. Streptomyces venezuelae produces nice single colonies on our nutrient agar streak plates after 2 days of growth. The plates were incubated for a further 3 days at 30 degrees. The colonies grew larger and started to sporulate, even on nutrient agar. M1582 has been genetically modified to overexpress the chloramphenicol biosynthesis genes. You can read more about this in Fernandez-Martinez et al. 2014. The genetic modification of M1582 makes it resistant to the antibiotic hygromycin, as can be seen by growth of M1582 but not wild type on the plate on the right. Now let's have a look at our MYM spore plates. Growth has started after 1 day at 30 degrees on both plates. As seen by the change of colour to bright white, wild type has begun to sporulate after 3 days. M1582 takes a bit longer. For our agar plug diffusion assays, we are going to make lawns of 3 indicator bacteria. We will plate 100 microliters of an overnight liquid culture of E. coli DS941, a bacterial strain resistant to streptomycin, DS941 containing the plasmid PSB1C3, which makes it resistant to chloramphenicol, and the gram-positive bacteria Micrococcus luteus. Before the bacteria dry into the plate, we spread them uniformly over the plate with a sterile glass spreader, or in the lab you will probably use a plastic disposable spreader. I use the broad end of a plastic sterile P1000 tip to cut discs in the Streptomyces venezuelae lawns. We can also use a metal cork borer for this operation. Remember to use a fresh plastic tip or to sterilize your cork borer for each spore plate. We now use the blunt forceps to transfer our discs from the spore plates onto our bacterial lawn. Ideally, the forceps will be sterilized between transferring each disc. Wild-type disks have been transferred onto all three lawns, so we can now make a start on the M1582 disks. Finally, we want to use filter paper circles infiltrated with chloramphenicol as a positive control. We take 5 microliters of 25 milligrams per mil chloramphenicol in acetone and spot onto filter paper discs. The acetone quickly dries, leaving behind the antibiotic. The filter paper discs are placed on our 3 indicator lawns using sterile, sharp forceps. The plates are moved carefully to the incubators and incubated for 2 days. E. coli at 37 degrees, Micrococcus luteus at 30 degrees. Plates are incubated without turning them upside down.